Hello everyone, I'm Muzafar Riyaz. I'm working as a PhD research scholar at the Entomology Research Institute, Loyola College, Chennai, India. Today, I will be sharing with you the work which I have been carried out so far in my PhD program since 2019. My work is based on the comparative and deep level phylogenetic analysis of superfamily Noctuoidea, Insecta, Lepidoptera using complete mitochondrial genome sequences and their phylogenetic implications with other Lepidopterans. First, let us start with a brief introduction about the moths. As we all know that moths are the close relatives of butterflies due to their similitude in shape and structure. The order Lepidoptera is divided into two divisions, division Heterocera, which includes the moths, and the division Rhaphelocera, which includes the butterflies. Moths, as you know, are the insects that are usually unseen and unloved just because most of the moths and their caterpillars are the pests of many vegetable and fruit crops. And due to their nocturnal behavior, as most of the moths fly during the night hours. Moths are the unsung heroes of the animal kingdom, considering their rich diversity and ecosystem services. Moths also take part in the pollination, as they pollinate many flowering plants, like most of the bees, flies, and other pollinating insects do. Besides pollination, moths are the diet of many of the birds, lizards, spiders, and a whole range of other animals as well, which makes them an extremely important part of our ecosystems. Their significant roles played by the moths in our ecosystems makes them a remarkable species in the animal kingdom. So talking about the classification, as we all know that classification is an important aspect in taxonomy, systematics, and evolutionary history of an organism. So to address some taxonomical, systematical, and evolutionary questions, we have taken the studies which are based on the complete mitochondrial genomes of superfamily Noctuoidea. The identification of all the species which have been carried out has already been done uh, based on the morphological characters. Now, this study which we are doing is the molecular study that will be a witness to the morphological identification, or using the DNA barcoding, which most of the taxonomists use to identify the moths or any other insect groups. So coming to the main topic, apart from playing significant role in pathways that are within the mitochondrion for producing energy, as we all know that the mitochondria are the powerhouse of the cells, the mitochondrial DNA plays a very crucial role in ecology and evolution of animal species. The mitochondrial genomes or the mitochondrial DNA is a small circular DNA comprising of 15,000 to 16,500 base pairs. The mitochondrial DNA is an excellent molecular marker as well, which we use in the studies of comparative and evolutionary genomics, molecular evolution, phylogenetics, and the population genetics of animals. Mitochondrial genomes have been widely used to address phylogenetic questions in invertebrates, particularly in insects, which outnumber every other organism in the planet Earth. And just because they have a huge population, huge number of families, orders, subfamilies, tribes, just because such a huge population is a very it's a very complicated kind of work which we carry out both in taxonomical aspect or evolutionary aspect or phylogenetic aspect. So coming to the mitochondrial genome, which is a compact structure, extremely conserved, maternally inherited, lacking genetic recombination, and having a relatively evolutionarily fast rate. So that's what we are deploying in this kind of work to address some of the questions in taxonomy as well as the systematics and in the evolutionary aspect as well. So coming to the insect mitochondrial genome, the insect mitochondrial genome are commonly circular with about 14 to 19 kilobase long, which contains 37 genes. The insect mitochondrial genome comprises of 37 genes. which includes 13 protein coding genes, 2 ATPase genes, ATP6 and ATP8, 3 cytochrome C oxidase genes, 1 to 3, 1 cytochrome B gene, 7 NADH dehydrogenase genes, NAD1 to 6 and NAD4L, 2 ribosomal RNA genes, RRNL and RRNS gene, 22 transfer tRNA genes, and an adenine A plus T rich region, which comprises the initiation sites for transcription and replication. This is the circular map of Hypospila bolinitis and complete mitochondrial genome, which is 15,640 base pairs long. The red portion comprises of RNA genes. The yellow portion comprises of NADH genes. The light pink comprises of cytochrome oxidase genes. The light green contains ATP8 and ATP6. The blue ones comprises of tRNA genes. Objectives of the study. The first objective of the study is to sequence the complete mitochondrial genome of different Noctuoidea species and finding the perfect gene arrangement. To sequence, annotate, characterize complete mitochondrial genome of Noctuoidea moths and to compare with other lepidopterans. We have selected the superfamily Noctuoidea for this study. To observe gene organization such as the gene locations, the start and stop codons in the particular genes, to compare the mitochondrial gene arrangement of the geographical population in the same species, which exist with different gene arrangement of outgroups. To discuss the mechanism of the gene arrangement in different geographical population or sibling species at mitochondrial gene level. Most important aspect of the study is to analyze the phylogenetic relationships of moth species among the selected superfamilies like Noctuoidea and outgroups which we select randomly from any other order like Hymenoptera, Orthoptera, or Odonata or Diptera. After that, the phylogenetic relationship will be analyzed based on the outcomes from the maximum likelihood method, which we used in the Mega software, and the Bayesian interference using the software packages like Mr. Bayes. Coming to the methodology, first things first, we collect the species and we select the species for mitochondrial gene analysis. Before that, we preserve the species, the sample in the 100% ethanol at -80 degrees Celsius until DNA extraction. After that, if we have to select it for the genomic analysis, we cut a small proportion of thorax for genomic DNA extraction. And in genomic DNA extraction, we use zymoquick DNA tissue and Insecta mini prep kit. After that, the quantity and quality of the mitochondrial genome is checked using nanodrop 2000 spectrophotometer, thermoscientific USA, qubit DNA HSA kit, thermoscientific USA on agrogel electrophoresis. After that, the whole genome sequence library will be prepared with Illumina compatible Netflix rapid DNA sequence bundle, Bioscientific USA at Genomic Technology Private Limited, Bangalore, and Molasses Scientific Limited, Bangalore, India. After that, it's time for the sequence assembly and the gene annotations and the phylogenetic analysis after that. So far, we have sequenced 36 moth species, the complete mitochondrial genomes of 36 moth species we have sequenced so far. We were having a target of 50 species, 50 to 60 species, but due to the lockdown, we stuck at the 36 species. In future, we will be completing the next 15 or 20 species as well. So here are the list of the species which we have sequenced so far, and all of the 36 species have been granted the accession numbers from the GenBank and CBI. So this is the length of the complete mitochondrial genome of the following species which I have sequenced so far. After that, so after getting the sequence assembled, the first step is to submit the complete mitogenome sequence of a particular moth species to GenBank and CBI for the accession numbers. Accession numbers are like the registration or license numbers for any species. So without accession numbers, it is the totally is not valuable at that level. If you are putting it in the manuscripts without accession numbers, it won't be accepted. So after granting the accession numbers, the complete sequences are submitted to the MITOS2 software for getting the perfect annotations. Here I will show you. So this table we got from the Mitos2 software. So in that software, we use it to submit all the genes, and this software is giving us this table. So the gene order is like this. One second. So this is the gene order, like NAD4L, tRNA genes, cytochrome B oxidase gene, tRNM genes like NAD genes, ATP8 genes. So this is the gene order A plus T region, and it's giving us the perfect strands. Also, the gene is present in the minus strand or plus strand, the location of the genes, the length of the genes, the anticodons if present in the genes, the start codons, the stop codons, and the intergenic nucleotides. After that, we used to segregate the genes like NADA genes. We segregate them in one file, then TRN genes we segregate them in other file for future uses. And after that, the gene arrangement or rearrangement. If these genes are having or have to do a little gene arrangement or rearrangement, we can do it here. After that, what we do is, first we sequence the annotation in Mitos2 software and Chlorobox software. After that, we calculate the skewness, AT skewness and GC skewness in the software vector builder tool. And the formula we used is for AT skew is A minus T divided by A plus T, like A content and T content in the protein coding genes. We use it the software vector builder tool for this. After that, the circular form which we construct in the OGDRA organelle genome maps, which we use the software, the Chlorobox. The tandem repeats finder tool, which we find the tandem repeats in any genes. After that, the phylogenetic tree construction, we use the Mega software and the Mr. D software. Then phylogenetic tree analysis, we use the fig tree for construct the trees. So here you can use the relative synonymous codon usage, the codons which are used by the tRNA genes. For example, this is the species Rusicata privata, Ischia manlia, Helvicorpa armigera, and this is the legend of the codons which are used by the particular tRNA genes. After that, what we do is the genomic organization and base composition. After that, the protein coding genes and the codon usage. If there is overlapping and intergenic spacers, we also calculate that also, and transfer genes, the ribosomal genes and the A plus T region. What we do is we find that the underlying genes are the tandem repeats, and these blue and pinks are the microsatellites, and this is the polystretch T. And here in case the microsatellites are there, and in case of motifs and polystretch T and polystretch A, which is present in the A plus T region of any mitochondrial genome sequence, motifs ATAGA we cover within it, and the polystretch T, then polystretch A. And in the tRNA genes, what we do is we search for any mismatching base pairs in the tRNA clover leaf structures. This is the acceptor region, the acceptor stem, anticodon loop. If there's any mismatch, we used to find it here. So after all this work, the next part and the main part and the conclusion of this study is to carry out the phylogenetic analysis. The phylogenetic analysis was employed using the sequence of 13 protein coding genes and two rRNA genes, RNL and RNS. For phylogenetic analysis, we used only 13 protein coding genes and two RNA genes. Each of the 13 protein coding genes or RNA genes were aligned and conjugated using Clustal W in MegaX software. In addition, concatenated amino acid sequence from 13 PCGs and RNA genes were used for reconstructing the phylogenetic tree, which was done using the MegaX program using the model-based maximum likelihood technique. So based on the 1,000 bootstrap replicates, the suitable model general reversible mitochondrial empty reverse gamma distributed with invariant sites. G plus I was to infer evolutionary relationships in the Unipro Eugene software. The map used to align amino acid sequence and conjugated the 13 protein coding genes and 2 RNA genes. So after all this process, the final step is the phylogenetic reconstruction, which you can see here is the phylogenetic reconstruction. So here what we do is we use it to look after the clades. These are the clades, different clades, and this is the bootstrap value, which you can't see in there because it's too small. So if a bootstrap proportion value is 70 to 100, so it's a perfect for the results. The outcomes will be the perfect. So after that, we use it to draw in the Microsoft Paint for aligning the perfect families and orders and superfamilies. Sorry, not orders, families, superfamilies, and if we use the tribes, we can also use that. So here you can see this is the alignment of the families like Arginine, Herminine, Erebine. So these are the families, Limentrine. So likewise, we use the phylogenetic tree analysis for 13 protein coding genes and 2 RNA genes to address some of the problems in the taxonomic positions in many species as well. So this tree is from the MegaX software. And another part of the phylogenetic analysis, we used the Mr. Bayesian analysis, Bayesian interference, we used to say also. So in the Bayesian interference, the data set was subject to the Bayesian interference BI analysis. The GTR plus IR plus R model was used in the BI analysis, and gamma rate variation between sites presented and performed using the program Mr. Bayes 3.2.7a version of the software. After that, the standard deviation of the split frequency showed that the MCMC convergence had reached below 0.01. So 0.01 is the average of this chain, Markov chain. If it is higher than 0.01, then the results will get deflected a little bit. With the data set of 4 chains, 3 hot and cold chain, we were unfound 2 million generations, with the tree being sampled every 1,000 generations. After that, the tree was constructed using the fig tree software. And you can see here also we have formatted the families based on the clades and the Bayesian interference values. So after that, what we do is we compare both the trees, which we get from the Mega software and the Bayesian interference. And if the results are similar, then we carry on forward. So in this software, this is a very complicated kind of work. This Bayesian Mr. Bayes software, it usually it is if we run it for 1,000 generations for 2 million, 1,000 generations for 2 million generations, it usually take 30 to 40 days to get finished in the laptop which we are using. And we don't have to shut down it also. We have to hibernate the laptop for continue running the software. So after that, so this is the first paper which came out from the study, the comparative mitochondrial genome analysis of Eudosima salamina, grammar, Lepidoptera novel gene arrangement, and phylogenetic relationship within the superfamily Noctuoidea. So this was the species Eudosima salamina which we first published. So all these annotations and the gene works have been completed yet for most of the species. And some of the species, 2 species which we submitted to the Zootexa, 4 species submitted to the scientific reports, 2 species submitted to the organisms diversity and evolution Springer journal and frontiers in ecology and evolution. And the same things will be going to the other species as well. So besides this work, I have been working on the insect diversity of Kashmir, the northwestern Himalayas. So I have been collecting the insects from that region also during the lockdown period. So in that lockdown period, I collected the species which are in the blue color. So these 2 species, the first 2 species are the new record of India, And the other three species which are in the blue color are the new records of are the new records from the Jammu and Kashmir Union Territory. And these are the publications I have been published in during the lockdown period. And these are some of the book chapters which I have published during the lockdown period as well. So at last, I would like to thank the Asian Council of Science Editors for giving me an opportunity to present my PhD work. And I would like to thank the Academy of Excellence in Biosciences for sponsoring this for sponsoring my registration and this presentation. I also like to thank the India SRB DST for funding this project, and I would also like to thank my host institution, Entomology Research Institute, Loyola College, Chennai, India, for always guiding and supporting me and providing the basic lab facilities. So in this way, I would like to conclude this presentation. I hope you have enjoyed it. Thank you very much.